This is CheloniaCast, episode 30. Today we are actually recording live at Rainbow Springs State Park, where we're attending the Rainbow Springs Turtle Survey. We're joined right now by Dr. Peter Meylan, who is Professor Emeritus in the Collegium of Natural Sciences at Eckerd College. Dr. Meylan has done this work at Rainbow Springs for over 30 years now and has done a lot of work with the ecology of Florida turtles. He also wrote the book on the biology and conservation of Florida turtles, but he's also done a lot of other turtle work and phylogenetics and other sort of disciplines. So we're really excited to talk to him today about some of his adventures and research experience.
All right, so Dr. Meylan, what first got you interested in turtles, and what made you decide to pursue Chelonian research, specifically in the areas of morphology and phylogenetics?
So I grew up in New Jersey, near the Passaic River, and when we were kids, we spent a lot of time in the woods and would find box turtles, spotted turtles, snapping turtles, painted turtles, wood turtles. I had a turtle pen in the backyard when I was probably in first and second grade. In the third grade, my mom signed me up for a herpetology course at it was then called the Morris Junior Museum in Morristown, New Jersey. And I was completely blown away by the fact that one could be a herpetologist. And so it was just downhill from there. So I worked for the museum, which became the Morris Museum of Arts and Sciences. I worked in their live animal room like all through high school. And then when it came time to apply for undergrad, there were going to be four of us in my family that would be in school at the same time, so I had to go to a state school. And I looked at University of Kansas, University of Arizona, and University of Florida because of herp programs. University of Florida was the last to accept me, and I had this weird logic, like they just made space for me at the last minute. So I came to Florida, and then right away I started volunteering in the Museum of Natural History. So what was then called the Florida State Museum, and they now called it the Florida Museum of Natural History. And then as a sophomore, I became the collections manager. And the curator was a guy named Walt Offenberg. Most people know of him from Komodo dragon studies. But he's done a lot of turtle work. And I actually started working with fossil lizards and snakes. So my master's thesis is on Pleistocene lizards and snakes, so that was, down the morphology line. And then Offenberg got an NSF grant to study land tortoises in East Africa, and he sent me to Africa to study land tortoises. And we did some of that together. We published a couple of papers, but I found that the softshell turtles were just a whole lot more interesting than
They're fascinating.
yeah, and their morphology is a lot more variable. So tortoise evolution is about changes in the skull size and shape and changes in the shell size and shape, but there aren't as many characters that work really well for cladistics with testudinids compared to soft shells. So when I was supposed to be collecting fossil tortoises in Africa, I ended up collecting a lot of soft shell fossils. And then that required going back and looking at the living material and the literature. And I found that there's just really, really nice variation in the shell and the skull of soft shells that worked really well with cladistics. And I forget what year it was, but there was a ASIH meeting in Gainesville, and one of Max Hecht's students, a guy named Meylan, I forget his first name, but he was doing cladistics with crocodilians. And I listened to his talk and I said, this is it, right? I was going to try to do fossil snakes, which Offenberg had done and was encouraging me to, to continue with. But I'm so glad that opportunity, that possibility of working with softshell turtles just popped up, and clearly it really helped me get to. It was that work that got me a postdoc at the American Museum of Natural History with Gaffney, right? Which is then when, I sort of went overboard into the morphology section.
The soft shells is a really interesting group, and what kind of got you for soft shells in particular? That was sort of a extension of work that Gaffney had been doing at the AMNH for all the different, I guess, clay families of turtles.
Yeah, but he hadn't done soft shells.
Yeah.
I mean, he had, by the time I got there, he had described a couple of soft shells, really good material that was at the American Museum that just needed to be named, but he hadn't done any real phylogenetics with a large suite of soft shells. And I think that it was, I think, Siebenrock's papers on the shell. So I had to learn German. I took graduate reading German so that I could
do this.
so I could get started with that.
Oh,
and then Siebenrock, and there's a guy named Heude, Heude, who had described variation.
The raft.
This is a story in anatomy. You think you discover something in anatomy, and it turns out some German already described it 100 years ago. So, and then in terms of the variation in the skull, Gaffney had just written this comparative cranial morphology of fossil and recent turtles. So it's in the same vein as some of Sam McDowell's papers. So McDowell was a famous morphologist who worked on deep systematics, doing dissections of musculature of heads of snakes. I mean, it's just really serious stuff. But I was able to use Gaffney's work, that comparative cranial morphology, to basically figure out where the variation might be in the skull. So if you look at that, the paper that's the bulletin of the American Museum of Phylogeny and Softshell Turtles, there's a section on the skull, non-shell postcrania, and then the shell. And all three data sets are really informative. So soft-shelled turtles have this incredible hyoid apparatus, right? So Chytra and Pellicelles especially, they're these suck gate feeders. But even these guys here, so the ferox that we catch here in Rainbow Run, they have a giant hyoid, like a snapping turtle, and their patterns, different patterns of ossification of the bones in the hyoid, that's phylogenetically informative. The vertebrae, maybe a little less. The lower jaw I did separately from the skull, but between those data sets, stuff came out, and then later on, a guy that was a student at Eckerd, Tag Engstrom, went and worked with Schaefer out at Berkeley, and they did a molecular phylogeny and came up with basically the same groups that I came up with. So it was really nice to see that the morphology held up.
And the total characters you were working with was well over 100, right?
Right.
So this is all very...
Right.
Yeah, that's a fascinating thing. Some of the characters that you look at, too, are sort of even intracranial almost.
Oh, yeah.
So the pterygoid and how that's... What are sort of the functional significance of differences there? Is that to do with musculature, or why would that be something that was phylogenetically informative for...
One of the things that I think was sort of a constant consideration in the early days of doing phylogenetic systematics was not to try to figure out why, right? Because you can make up stories about why. But the fact is, the morphology, it is what I see, right? I see the morphology. And I used to debate with Pritchard about this, about he'd get all into about, the function of a long neck. It's like, it doesn't matter what it, why it's there. I mean, if all chelids have the same arrangement of the cervical articulations of the neck, why that happened or how it happened, you will never know. You may never know. But here's some morphology. We can get out all these. We can get out the vertebral columns, the cervical vertebrae for all these chelids, and look at them, and there it is. There's the pattern, right? There's no debate about that. So I think sometimes, I mean, certainly in those days, and I don't know if it's still the case. Well, do people worry about how the different changes in gene sequences affect organisms before they use a sequence to say, oh, well, these guys are more closely related? It's like, I'm not quite, in some cases, it'll be really cool to know what those genes do. And there, I think you probably can make a strong argument for what the selective value might have been at the time, changes took place. But
that's sort of a whole different sort of analysis that we're not even sort of the cutting edge there is trying to figure out what these genes actually do. And that's a good point. I mean, genetics itself is kind of just marketed as a more robust version of morphological cladistics, essentially, is you're just looking at how different letter sequences are
right.
different.
No, I'm sure that that the morphological folks working with phylogenetics, systematics, and morphology set the stage for genetic data sets to be used in phylogeny reconstruction. And there's still a lot of good stuff being done. I mean, I'm not doing that stuff anymore because Annie and I have gotten so busy with sea turtles, right, that I had to quit doing something.
Right.
And so I have like one or two papers still in the mill dealing with morphology, but it's not going anywhere. And the sea turtle stuff is really pressing because there are conservation issues that need to be dealt with right away.
Maybe we could talk a little about the sea turtle work. That's sort of a unique aspect. A lot of freshwater turtle ecologists don't really dabble in the sea turtles as much. So, and you've sort of done that, but you've done a lot of work in Bermuda, I know. Maybe you could
both Bermuda and Panama.
Okay.
So when when we were graduate students, Annie did her PhD thesis in Bocas del Toro, Panama, on foraging biology of hawksbill turtles. And together we did a bunch of what were called West Atlantic surveys, where we went to different parts of the Caribbean to see what was going on with sea turtles, just by talking to fishermen. So Archie Carr got us involved in that, and then also got us involved in work in Bermuda. And it really does come down to morphology, because in the late 70s, we learned to do laparoscopy. So it's a minor surgical procedure where you can look at gonads. And we did it in Panama starting, 1989 would have been the first year, and then starting in Bermuda, 1990. And at that time, there was this question about developmental habitat. Are there places where young sea turtles go to grow up where there are no adults? And it turned out we could test that in Panama, and we could test it in Bermuda using laparoscopy. So we did, it was kind of like where you guys are now, or maybe a little bit later, where you have lots of time, no kids, no tenure track jobs, and we could come up with just enough money that we could travel every year to Bermuda and Panama. And then in between, I was, traveling with Gaffney to go collect, data. He always said, the best fossils are in museums. There's no point in going out and trying to dig them up. There are tons of really great fossils in museums. So we spent a lot of time going to European museums, went to museums in Brazil, and all over the U.S. looking at fossils. And I think it was probably when Annie and I had started to have kids, and the choice was, do we want to go and spend our summer in the bowels of some dark museum?
Yeah.
Or do we want to go to Bermuda and Panama, right?
Yeah.
So it was like the sea turtle slowly but surely got the upper hand. I mean, Gene and I had done so much work together for so many years that it was possible for me to keep, publishing with him on especially side neck turtles, in the later part of that. And, whenever, I don't do this anymore, I don't interview candidates to come teach at Eckerd College, but I would always advise candidates to keep in good stead with your postdoc advisor, and keep publishing with your postdoc advisor, because it's hard teaching at a place like Eckerd, right? You have a lot of teaching, a lot of mentoring, committee duties, and it's hard to come up with complete new projects on your own. But if you can keep working with your postdoc advisor,
well, we're doing. I mean, it's fine. It's fun.
A big snapper has just come in. You want me to come? Can we pause?
Yeah, let's
go
pause
get the snapping turtle and come back.
All right, we'll be back in a second.
Yes, we've just had a little interruption.
Yeah.
So when we catch big turtles here on the Rainbow River, sometimes it's best to get them out of the boat so that they don't have to worry about piling smaller turtles in with bigger turtles that could possibly damage the capture. So we have a snapping turtle and a big soft shell that have just come into the pavilion where we work up turtles.
It was exciting to watch too. One of the students sprinted down here. He's like, "We got it."
We should have looked to see what else they had in the boat. I didn't even think to ask, how they're doing.
Yeah.
But by now they'll probably have about 50 turtles in that boat already. Yeah.
Maybe we could even. You were talking about a little bit about how I was curious, kind of how you do both kind of ecology and then the systematics and the phylogenetic side. It's not something you commonly see. I mean, a lot of it seems like.
I agree 100% that people end up getting in channels and then have trouble, diversifying. And I think I was really lucky to be in a vein where I ended up, doing different areas, and it's just fortuitous. I think there was one of the questions that y'all were interested in is how did I decide to do a long-term study at Rainbow Run? And I didn't decide. I mean, we started doing it, and then, after a while, it kind of slacked off, and students came back and said, "Hey, how come we're not doing more of this? Let's go back and do it some more." And then, this is the 114th sample today. Starting from 1990, and it's just a really great data set. We haven't done enough with it yet, but I'm hoping with having Jeff Gessling and Peter Scott at Eckerd College, along with students do senior theses, and
we got it. Sorry, I get a break for it. I just saw him
snapping turtles escaping.
We'll keep it.
Put him back in the tub, and then just set the other tub on top. But we'll still have to keep an eye on him.
This is great, though. All right, we'll run back. We don't need to stop.
All right, let's get back at it.
All right. Well, let's go to live sample.
Right.
Yeah.
Yeah. So anyway, with additional faculty now at Eckerd College that are physiological ecologists in the case of Gessling, and population geneticist and ecologist in the case of Peter Scott. I mean, it's just too good, that this can go on, and I think that having the long-term data set and seeing what's going on, with freshwater turtles in one place over a long period of time. And the really cool thing is that there is a study by a guy named Marchand, who was a student of Archie Carr's. He actually went into dentistry. He was my dentist when I was when I was undergrad.
I wondered where he ended up.
So this guy Louis Marchand, who actually I think Carr and Marchand named Graptomys barberi.
That's right.
That's why you know that name. Yeah. So Marchand did his master's thesis in here and collected the data in the late 1940s. So that's why we know that Sternotheris minor was not in Rainbow Run in the 1940s, and somebody brought it here. And I think that Peter Scott's going to be able to figure out where it came from.
Where they came from?
Yeah.
Wow.
Yeah.
I'm interested to see if they resemble the Santa Fe ones. That might be some clues, but then again,
But genetically,
Yeah.
I mean, it's going to that's going to
That's
be
right.
it. It's very possible that they came from Silver Springs. So.
That's really
Yeah,
interesting.
Paul Mulder knows who it is that apparently brought them here. And I think that at some point we have to make sure all that gets written down.
Well, and that was in the Carr and Marchand paper. Species composition has changed a lot. Like you said, that's been something that the minor have just taken off, right?
Oh, yeah.
That's
Well, and now I don't know if you heard, I was talking to Jerry right when we were doing introductions. Our last sample, there were actually more Consina than minor. So Consina really is a river turtle, and spring runs with clear water are just ideal Consina habitat because they can forage all the time. This is why Jerry's turtles in the Santa Fe, they go into the springs when the river clouds up. So they're moving up and down the river all the time. And here the turtles don't have to move, right? They have 7 miles of clear spring run where they can, just anywhere you look, there's forage for these guys. So.
And this system itself is kind of interesting. It's not super close to the coast, but it just feeds into the Rainbow River, and then that's kind of a...
So Rainbow Run or Rainbow River, right? So a run is spring run. We do this run, and people think it's like we're going jogging or something. It's weird. Anyway, a spring run, like Hitchtuckney Spring Run, is a kind of a river. And the Rainbow River flows into the Withlacoochee River, and then the Withlacoochee River flows into the Gulf of Mexico down near Crystal River. But this coast of Florida and a lot of Florida, you have so many of these really nice springs, and I hope that people appreciate them. Floridians love them, but we kind of love them to death. Mm-hmm. So, at Hitchtuckney, they have a limit on how many people can go down the river every day. As far as I know, there are no limits here. We should talk to Jeff Sowards, the aquatic refuge guy who's here today, about, what are the ideas about limiting how big a boat motor can I have on this river, or how many people per day. Eventually that'll have to happen. Just the amount of development in this part of Florida is just incredible. And so far, it seems like the turtles in this river aren't suffering for it, but certainly other turtles like gopher tortoises are in bad shape because of the rate of development here in Florida.
So there's definitely some anthropogenic sort of stressors, obviously on this area. Mm-hmm. One thing too that was curious, I think in one of the papers on the Rainbow on this system that it was published, you mentioned that over time the cooter size changed, the average size, and maybe that was due to impacts of boat strikes or something.
So generally speaking, there are now a lot more adult-sized cooters than historically, and I think the really serious issue that existed probably when we first got started here was that people were coming here to get turtles to eat, and I've made this point, many times, and in meetings with the Game Commission, that when people go to get turtles to eat, if they're getting Pseudemys, the sexual size dimorphism is a real disadvantage for those cooters, because what they end up doing is taking the reproductive females. And that's, I mean, it's like what people did with sea turtles, taking the nesting females off the beaches, right? So it's the same thing where you're removing the laying hens. And I'm thinking that probably stopped maybe 20 years ago. And it's just taken additional time to get large numbers of reproductive size, especially Floridana and Consina. So the two big Pseudemys that are here, and we should see today. I mean, you'll see some 7 and 8, and maybe even a 10-kilo Consina female, and I think that's the reason why. Last time we were here, we caught more Consina than minor, because there are lots of Consina that are 2 and 3 and 4 and 5 years old.
Are there any ideas in terms of the population size of some of the turtles in here? And if you're talking that there definitely are. Some of the minor populations, it's pretty massive, but is that the same case here?
I've tried to do estimates of the population size. So we did one in that very first paper, in the Florida Scientist paper, and I forget the numbers right now. But we've tried to look at the population size of minor, but it's always we just do a 2-day sample. So we sample one day and come back the next day, and so it's the numbers I don't think are very reliable. But the other thing that we have been doing is to pit tag all of the adult female minor, with the idea that at some point somebody's going to do a life table for minor. Right. So we did estimates for the Pseudemys in a paper written. So Diana Hustis was an Eckerd student we did a paper on Pseudemys that was pretty detailed. But like I said, having these different projects, Panama, Bermuda, doing stuff with Gaffney, I haven't done with the Rainbow Run data, everything that could be done. And
That's interesting.
But now that I'm retired, I can get to work.
Yeah.
This is a little bit unrelated, but Pritchard had cited you as recording the largest Florida softshell turtle on record, if I remember correctly. Or you reported that it was 96 pounds, came from like Okeechobee. What was the story with that?
I think it's in that Florida turtle volume, and which I edited, by the way. I didn't write it, right? So I coerced people into writing the chapters. It's from Lake Okeechobee, where one of the rivers goes into Lake Okeechobee, and I thought that it was Pritchard himself who had gotten maybe the skull from it or something like that.
He said he has the skeletal remains of a slightly smaller one, but that big one might be at UF or something, is what I heard. But
I don't remember all the details.
About if someone had caught it or what the story was.
There's so much stuff on the hard drive that it's hard to find it all.
Yeah.
The Florida turtle, I guess compilation that you brought together. That's a fascinating piece.
Yeah.
I guess what were some of the things that you learned from that? I'm curious because you didn't write all the chapters obviously, but you put it together, so there must have been some cool, I guess, tidbits of knowledge maybe that were picked up through doing that. Or what was that process like? I guess.
Well, it was really great, and I don't know if y'all have talked to George Heinrichs yet.
A little bit.
Know him? Yeah. So George was really the one who got me interested in the idea of getting turtle biologists together in Florida, and we had a couple of meetings at Eckerd, maybe in the 80s and 90s, and then at the turn of the millennium we were doing it again, and we had this idea that there's enough expertise that we could get a book together on what's going on with Florida turtles. So George was a huge influence in getting that whole thing started. And then I think that it was from my days as a collections manager in Gainesville at the Florida State Museum. So I did that for 11 years, and I got to know lots and lots of herpetologists. So people like Dale Jackson and Paul Moeller and other people, Pritchard, people that were doing different turtle studies. And then through Annie, I knew the sea turtle people. So that was like 5 species right there. And then John Iverson has always been a great buddy of mine. It sort of came together. We did a little work here and there to fill in where somebody said originally, well, I'll do this chapter, and then they didn't, and we could find somebody. But it was great to like you say, bring all that together. And then in terms of like the most interesting things that I learned, there are 2 turtles in Florida that make really remarkable use of ephemeral wetlands, and I don't think we appreciate that enough. I mean, people think about amphibians using ephemeral wetlands. So the chicken turtle and the spotted turtle both use ephemeral wetlands, and the strategies are completely different. The chicken turtle, when the ponds dry up, the chicken turtles go in the woods, and the spotted turtles go to permanent wetlands. So, for spotted turtle survival, you need, wetland mosaics, because they come out of the permanent wetlands and into these ephemeral wetlands because the resources are so incredible. And then the chicken turtles, I mean, the folks at SREL, and I guess Buellman figured this out originally in Virginia, that chicken turtles, they get out and go into woods and hide out, wait for the ponds to fill up again. And I learned from doing those trips with Iverson, going to look for mud turtles all over, how important ephemeral wetlands are for certain kinds of turtles. So yellow mud turtles are amazing, right? They'll be underground for most of the year. Ponds in the Sonoran Desert fill up. They all come out. They eat whatever invertebrates and tadpoles, they gorge for a couple of weeks, they mate, and they disappear again.
Yeah.
Right? So it's just, it's an amazing thing that turtles can do. And I think that figuring out that there were turtles that were using ephemeral wetlands in a big way in Florida is really important. It's also really important in terms of conservation, right? You got to know the biology if you're going to do any kind of effective conservation. And then the other one that I'll mention is one that, so in the book, we push the idea that the mud turtle, so Kinosternum subrubrum in peninsular Florida, really is a different beast. And so now it's Kinosternum steindockneri. We don't know squat about it. We don't know. Yeah. We've never seen. We've caught Kinosternum boweri. We've caught gopher tortoises. We've caught Dirocheles. We've never caught a steindockneri here, but they're here. I think in most cases, you look at the data and people find them crossing the road. You catch one in a trap one time, but I don't know any place where people have a marked population of steindockneri and can say, this is steindockneri habitat. It's like steindockneri are where you find them. And I think that for a long time, spotted turtles were that way, right? Spotted turtles were where people found them crossing roads, but now there are people that have done telemetry of spotted turtles in Florida and Georgia, and people are figuring out, where they go and what they do.
Jack's used to the spotted turtles from the Northeast.
Yeah, the Northeast is not a problem, but
yeah.
I mean, a couple of guys have recently done a lot with them down here.
Right.
I've seen up in down into the northern part of the peninsula, but with the steindockneri, there's almost nothing. Anyone I know that's trapped them or looked for them, you get one. And I've trapped bari, and you can get a ton of them consistently, easily.
Yeah.
But no, it's never like that with steindockneri. It's like you get one every 10 trap nights or something crazy like that.
There's one location, and I kind of know where this is. I'm not going to say exactly, but someone has I guess most recent TSA meeting, they hypothesized that what they do is, for the most part, not even for estivation or anything. They just, when they're not feeding, they bury significantly deep within the mud, and they just come out for really short periods of time when rain fills up
Right.
these puddles. And that there is a
So it could be
population.
another turtle that's using ephemeral wetlands
Right,
in a
yeah.
different way.
I think so.
Yeah.
I mean, it seems like it. Even like, right, yeah, ephemeral wetlands. People see them crossing roads. That's when it's raining, if there's any correlation there. But they're fascinating. It kind of remind me of the Claudius.
Yeah.
That's a time of Elise, the beaks and just the ferociousness.
Well, and they're not. I mean, the defining characteristics are all have to do with reduction of the plastron, which would suggest that terrestriality is. I mean, kinos that are really terrestrial are box turtles,
Yeah.
right? So there's some that can seal up just as well as a box turtle,
Right.
And
Right.
yeah, and Steindochnerai is doing something very different.
It's more reduced, right?
Exactly. Yeah.
So
Very reduced plastron. Yeah.
that's a really interesting sort of scenario there. I guess with the it must be really an interesting thing to see. How long did it take sort of to put together the Florida turtle book? It was a long process.
Yeah. No. We
Yeah.
it was 6 years because we said, it's the turn of the millennium. We need this book. That was 2000, and the publication date is 2006. So it was like, George got together a huge collection of photographs, and part of it is we have lots of really nice photographs, and he was able to talk to a lot of different people. This guy, Dick Bartlett, for example,
Yeah.
gave us a lot of nice. I mean, everybody just donated their best pictures. And then Jamie Barichevich did a lot of work on the maps. And then, I sent everything out to be reviewed by what I thought were peer reviewers. I thought we had plenty of reviews, and then when Anders got it, he sent everything out again for more reviews. So I think what came out of it is really, really good. It might not be exactly what some people were hoping for. I think some people were hoping for a book that would be more for the general public. But I think it's a book that will stand the test of time. That it documents what we knew about Florida turtles, in the year 2000.
Yeah, it's a good compilation, and I like the fact that public can access it. It just takes a little more work, right?
Right.
But you mentioned trips with Dr. Iverson. That sounds really interesting, and to look for the kind of sternids.
Yeah.
What
So he's the person who brought me to Rainbow Run.
Oh wow.
So the reason we're here today is because of John Iverson.
Oh
So I started as an undergraduate at UF when John started as a graduate student, and his master's thesis, for which Peter Scott sings the praises,
right.
is a geographic variation in Sternothrus minor. And Peter Scott says, Iverson did a great job with that paper. But to do that paper, I had to skip school every Friday and go to a different spring run. So I think I had to do Calc 2 twice because of John Iverson, right? Because we were always off going to some, and I think I've probably been in most every spring, certainly in the northern peninsular Florida. I'm sure Paul Moeller's been in more springs than I have. But we would go and collect Sternothrus minor, and that was in the days when people were still collecting. You wouldn't think anything of going and catching a bunch of turtles and pickling them. And people were doing it with graptomes, and we were doing it with Kinosternon in Mexico. We would get, all the permits you needed. We would get a special permit for the turtles, and then separate permits for for general herp collecting. And I think every year, I want to say maybe from about 73, 74 until the late 80s, I would go with John and other people. So a guy named Dieter Ojica from Argentina. So he's now an airline pilot, but he was our Spanish person. You always needed somebody with good Spanish.
Yeah.
And neither John nor I spoke Spanish. So Diderot used to go. A guy named Ron McGill, who still works for the Metro-Dade Zoo down in Miami.
Okay.
Cuban extraction. I'm trying to think who else went along as like the third person. One year we went with 2 vehicles and 6 people, and we drove all the way down to the Yucatan Peninsula and drove around. So Paul Molder was on that trip. Yeah, so I'm pretty sure I've been in every state in Mexico and have collected almost every. I know the one kind of sternin that I haven't collected is Creaseri, the one in the Yucatan Peninsula.
Oh, okay.
But I think everything else. Oh no, some of the new ones that Dick Vogt named that are in these little isolated drainages on the Pacific side of Mexico. We didn't get those, but no, I can just remember setting traps with John Iverson just in I think every little river in Belmont.
Not only have you been to every state, you've been to every
saltwater. And this was before Google Maps, right?
Right. Yeah.
But UF had a great map library, paper maps, and John would go to the map library and Xerox every topo map of the trail that we would go on. Right? He'd had the whole sampling regime scheduled, and you had a black book in which all the maps had been, all the maps of the places we were going, and the little reservoirs would be circled, right? And you would go, oh yeah, there's a place coming up on the right. It's like 2 miles up here on the right, and you could say, oh, it must be those trees out there, right? That has to be it.
Right.
Stop the car, throw in a couple of traps, and say, well, we're just a little bit up the road from the next little river. Why don't we'll go set some traps there, and then we'll come back and check these. And so you would do that, and inevitably, while you were doing this, some local person would say, are you guys like looking for snakes and stuff? Because there's a rattlesnake like right over there. And so we would just incidentally to doing the kind of sternin work, we saw lots and lots of really, really cool herps all over Mexico.
That's incredible.
Yeah, no, it was great.
And you have to put a little more effort into it. Nowadays, I feel like it's so easy to access different areas. It's almost like it's a little bit underappreciated for how tough it really was to get there and first discover those things and just analyze that. I mean, that's incredible to hear.
But I feel like for us traveling around Mexico, we took a lot less precaution than I think you would need to take now. I mean, we just, we would finish up at the end of the day and say, oh, this looks like a good spot. Why don't we camp here? And then the next morning, people would be walking by the tent, and you'd realize that you had camped in the trail that went from this little town to the nearest bus stop. And all these people are looking at you like, what the hell are you doing? Being in Main Street. Yeah.
I spent a little time in Belize this summer and at the Be Free. Jacob Marlin, who's the director there, was telling us about some trips he used to take, or he even recently driving to Belize, and you can't stop even in some of these spots. It's such a different time back then.
I can remember being in the car with John and this guy Dieter Ojica, and we were driving, I think, through maybe Oaxaca City, and a policeman blew his whistle and said, stop, stop. And Diderot said, don't stop, go. It's
a good philosophy. That's
No.
yeah. I'm curious too, like what because you've traveled a lot, like you said, the museums and that sort of thing. But what's been maybe like the pinnacle experience in terms of going out in the field, or favorite place that you've been to out of all of your pursuits of turtles?
The the trips to Mexico really stand out. When I was a sophomore in high school, I had read Carl Kaufeld's book Snakes and Snake Hunting, and I took the train to Jasper County, South Carolina, and the most fun I've ever had looking for snakes was doing that, going through recent longleaf pine burns in southernmost South Carolina and not knowing whether the next snake you see is going to be a canebrake or a copperhead or a king snake or a red rat snake or a rainbow snake. I mean, or there were a lot of people that were just focused on the snakes, but there's like maybe salamander. There's a pseudacris up there. I forget which one it is, but there's a local pseudacris that doesn't occur anywhere else. And at that time, you could still find Ambystomys cingulatum. I don't know if it's still there now. I remember picking up Dirocheles shells. There were mud turtles everywhere, right? Mud turtles crossing the road. Nobody, the people that were there, a lot of the other snake hunters, quote unquote snake hunters, were interested in finding diamondbacks and canebrakes. But I just enjoyed this huge diversity of herps. And that was one of the coolest trips. But then I'll say, from over the long haul, one of the most interesting things has been to raise two sons. So my boys are a little older than you guys, so 30 and 34. And maybe is that a lot older than you guys?
Yeah, we're 18.
Yeah. Okay. Yeah. So we grew up. They grew up going to Panama. Usually in July, May, June, July, we would go to Panama for a month. And then in August, we always went to Bermuda. And Panama, working in the field in Panama is, I mean, you're really remote. You're drinking water that you're catching off the roof. There's no connectivity. I mean, there is now a little bit. We used to just take piles of books for them to read. Then they would go to Bermuda, and Bermuda is like Switzerland or just it's a European place in the middle of the ocean. And in fact, my family is Swiss, and sometimes in between those two trips, we would go to Switzerland. So sort of the contrast of going, doing turtle work in a remote place and then doing it in a super civilized place. Just seeing that contrast year after year and watching my boys grow up, doing that was really cool.
That's cool. That real. I mean, it's a great learning experience to travel as a young person and see kind of a diversity. A lot of people in the United States kind of stay in one area, but that's cool.
Yeah, and I actually did. I made 3 trips to Africa during the time that I was working with Offenberg, and those trips. I'm always thinking I would just love to go back to. So I was at a place called Kubifora up on Lake Turkana, hunting fossils. So it was one place where I did go in the field and look for fossils. And I've described the softshell turtles, but never. There are a bunch of Pelusios fossils from up there, and I had always wanted to do with Pelusios what I did with softshell turtles. But it's a project, I borrowed specimens and had them for years and just finally just said, I'm not getting to this. I had to send all this stuff back. And it's one of my big regrets is never doing a morphological treatment of Pelusios, which is another really cool, a really cool turtle.
The Turkana, yeah, go ahead.
Oh, no, I was just thinking about the fossil side necks. And some of the skull diagrams in the paper were amazingly drawn, and it represented some incredible variation in cranial morphology that I had no idea existed.
No, I think that the diversification of side neck sea turtles is something that people had really missed up to the point where Gaffney, used his clout to beg, borrow, steal, get all of this material together, both for these Bothrymytids and then also for the Podocnomytid group, which includes maybe one or two sea turtle clades. But yeah, I mean, you have side neck turtles that do the loggerhead thing.
Mm-hmm.
They do the soft shell thing. There's one with a long, narrow snout.
Right. Yeah.
There's one that has a really round clipping snout like a green turtle.
Yeah.
So and then in terms of the actual illustrations that went into those publications, one of the really amazing assets of the American Museum of Natural History was this illustrators corps. And three people in particular. There's an old guy, Chester Tarka. He was like the kingpin. And then Lorraine Meeker. And then a guy named Frank Ippolito. And the three of them are responsible for the majority of the illustrations. They also had interns that also would come, learning to do scientific illustration. And I think, with digital photography, what it is, and different kinds of imaging that people can do now, maybe scientific illustration is getting really rare. But when you go back and look at some of those images, I mean, the nicest things from Gaffney's work is where you have an image and then you have a line drawing next to it, and the line drawing is telling you what you're seeing in the image, and gosh, the millions of dollars that the American Museum must have put into all of those illustrations. It just, it boggles the mind that all that got done, and Gene was just extremely good about making sure that all the illustrations were perfect, right, anatomically correct, and then kept organized. I mean, just the logistics of doing something like that big Bothrymytid monograph. It's just a huge amount of work. And it was just fantastic to be able to work with him over a lot of years.
It highlights one of the when I first saw that paper, it was incredible to see the diversity of the Pelamedusoides historically. I mean, you look at contemporarily, and there's, compared to historically, there's so few species within that group.
Right. It's basically Podocnemys is all that's left. And then, when you think about, there's a whole clade of what are very likely marine Podocnomytids, and they just turn up everywhere. I mean, they're in the Caribbean, they're in Cuba, and they're in Puerto Rico, and they're in South Carolina, and they're in Colombia. Then they're over, in Africa, so India. So they're worldwide, and so side neck turtles did as well as sea turtles, as modern chelonioids, and at least twice. So.
There's multiple radiations of
So Annie and I were just asked about. We did an introductory chapter for a sea turtle methods manual. So we did it's an introduction to evolution, life history, and biology of sea turtles. It's like 4 pages, and they're going to redo this volume. It's 25 years old now, and they've just asked us, would we contribute, do that introductory chapter again. And I'm looking forward to getting caught up on more of the work that's been done recently, that goes into the question of how many times have turtles gone marine,
Right. It's fascinating.
It's a bunch.
It's tough to really narrow it down too. Like when you think about the radiations, like you said, the I guess geographic diversity of the side neck turtles just historically is incredible, and actually trying to match that up with predictions of just how the physiographic changes that were occurring at the time. It's that's a whole task in itself. I mean, when it comes to diversity, what do you think exactly about that distribution? What does that say about the biogeography of the time, or is it?
I think it's a combination of plate movement, but then just the stochasticity of lineages blinking out versus other lineages, for whatever reason, hitting someplace at the right time that led to the kind of diversification that you see with, a lot of these Bothromyid species that Gene came up with and we described. They're all from like a relatively short period, in terms of turtle evolution, a relatively short period from Morocco. It's like one place, and this is where all these guys are. And so what was going on, I think it goes back to the question, how easy is it to figure out questions of why, right? Why did this happen? We know what happened, right? They're the bones. You can see the bones. but what was it that led to that? Boy, it's a lot harder. It's a lot harder a question to delve into. But I think, nowadays, more and more papers are being written by sets of people with different expertise. So you get some historical geologists to come work with the turtle biologists and can do a better job of sorting out the relationship of plate movements with why, like, Pararipimytids ended up on both sides of the Atlantic.
It's fascinating to think about just how stuff has changed so drastically over time, and we really have evolution by natural selection explains small variation, but we don't really have much of a basis to explain these large jumps over time. It's so interesting to think about that whole concept that drives so much of biology is really kind of, there's not a lot behind that. It's the history.
Well, and for folks that are interested in that, this guy Olivier Riepel, he's a developmental biologist mostly, but also a turtle guy. He recently published a book, Turtles as Hopeful Monsters. And it's sort of idiosyncratic, but it's worth a read. I mean, it really, I think he's on the cusp of something where it seems to be Evo Devo. So, this guy Max Hecht, who's a professor at Queens College in New York City. He used to come to the American Museum all the time when I was a postdoc there, and he always mentioned this fact that if you have a mutation very early in development, the implications are huge, right? And basically what Riepel is saying is that's how we got turtles. Right? You had this huge change that happened very early in development, right? And so I think turtles are a good example of a group that somebody with the right training could do what we were talking about just a little while ago about, does it really matter that what these genes are doing? But if you know what part of turtle development they control, and then you can see where they're shared among different groups of turtles. I mean, turtles have been around for so freaking long, right? So, Gaffney and I have always said, Pleurodires, cryptodires, they split into Triassic. Other people say, oh, no, you guys are wrong. But there's a good chance that pleurodires and cryptodires, that this thing called Protorocursus really is a pleurodire. It has a pelvis fused into the shell. So, and there's some difference, I can't remember right now, how many thoracic vertebrae, and it's got the right number to be a pleurodire and not a cryptodire. So the point is that you have living pleurodires and living cryptodires that you can get the genes from and say, oh, holy shit, this is the gene that makes the neck go sideways, or not.
I mean, yeah, technically, the morphology
and maybe, you can detect that comes from within cryptodires, or that it's a completely different set of genes in pleurodires or cryptodires that end up controlling the cervical articulations.
I mean, you should theoretically be able to back up morphological phylogenies with genetics, but it's just tough to say. Are we capturing all variation with two different data sets? And like, where do they overlap? It's sort of hard to.
Well, I think people have been doing it. Certainly there's agreement at the family level. The paper that Gene and I put together, a phylogeny of turtles. It's my most cited paper. And Gene and I basically decided one day, we're going to contribute a chapter to this volume by this guy, Michael Benton. Let's just get all the cladograms that we have and put them all in the same paper. And it was like a week. And we just whipped together this paper. And again, really good illustrators made the cladograms. It has a few figures in it. But it was like the first complete phylogeny of turtles. What we came up with in terms of living, where you can get the molecular data, those groups are the groups. I mean, we had this thing called Trionychoidea, where we were saying that Kinosternids are related to soft-shelled turtles. That was mostly my fault. And then the alternative was what E. Williams said was that, snapping turtles and Kinosternids belong together. And that's sort of maybe one of the bigger molecular disagreements with what we said based on morphology. But there's lots of stuff like, the fact that Dermatemes goes with Kinosternin, right? Mm-hmm. The Coreticheles and soft-shelled turtles going together. The Testudinoidea, comes out, So, yeah.
Yeah, that was, I went into the library and I've seen that paper cited a lot. I think we all have, but I went in and actually found the volume because I'd never been able to access it.
Mm-hmm.
It's going through. That's just fascinating. Like the intrafamilial relationships, but you also sort of place turtles in context of diapsids, synapsids, and that's changed, of course, but it was
right.
Well, I think we can wrap up because we're trying to get into the water. But for a last question,
I want to go catch a turtle.
Yeah, no, but we spent a long time talking about them. Now we need to go catch some. But I guess for a last question, sort of, if you had one major piece of advice for someone interested in getting into herpetology or turtles as a career, what would that be?
Go to a good college. Yeah, you have to get into a good program and then do the very best that you can. I always counsel students that if you can do it, do a math minor, right? Along with your bio major, your zoology major, whatever. So there's a student here today, an Eckerd student, Christina Lopez Miranda. So she's from Mexico City, and she worked for us in Panama this year, and she came to talk to me since she got back from Panama, and I got back from Panama as well. And It turns out she's taking Calc 3. And I'm like, this is it, right?
Nice.
You're golden, right?
Yeah.
Because she's really good. She's a good biologist to start with. And I think she's doing a chem minor and a math minor. So those are the kinds of things that will, I mean, there's just so many people that want to get into grad school. And I think the number of places in grad school is always limited. So having good academic credentials, but then also doing stuff like this, right? Having field experience on your repertoire. You don't know how many advertisements I see for students to help with sea turtle projects who can drive an ATV, right? So practical experience really does count for something. And when I write letters of recommendation, especially for students that have helped with this project, I always talk about, this is a kid that will show up, right, when you ask them to, and work hard all day, which not everybody with straight A's will do that, right? Mm-hmm. So being able to do field work is really good.
Mm-hmm. Yeah. I mean, that's we'll take that advice to heart.
And go get in the water.
Yeah. All right. Well, we'll do that. But
right.
That's going to conclude this season finale, I guess, is kind of where we're at. But thanks for tuning in, and we'll see you on the next one.
Good.
Yeah. All right. We're here with Dr. Peter Scott, who recently started a position as a I guess tell us
assistant professor at Eckerd College in St. Petersburg, Florida.
Cool. Yeah, we actually met back at one of the Desert Tortoise symposium meetings. I think it was an auction. I got some sea turtle books. I remember that was. But it's cool to meet up later on and sort of cross paths again. But I guess Dr. Scott can give us just a little overview of some of the stuff. We've been busy all day working up cooters and a lot of different species here at Rainbow Springs. But what kind of questions are we asking with these turtles?
So we're asking a number of questions. We're actually at Dr. Peter Meylan's kind of long-term study site. He's the one that established these turtle surveys, and what we're after are a number of things. Predominantly, we're after understanding long-term demographics and population trends that we hope we can turn into these things we call life tables, which give us ideas for how long each life stage lives and the proportion of survivorship and mortality those life stages suffer. And those are really, really useful when we think about conservation practices, because those are the numbers we want when we want to understand how our actions are potentially impacting populations, and they can kind of help inform how much extra stress populations can undergo before they might get extirpated or really start tanking.
That's a really interesting way to put a good overview. And that's all sort of the physical, like just actually tracking the turtles, recapturing. We can get those ideas through math, I guess, modeling.
Yep.
You're bringing sort of a genetics perspective to the table too. I guess do you have sort of questions for the future there? And
so some of the things we're thinking about with genetics is we kind of have two ways we think about populations. We think about a census size, which is how many animals are physically there. And then we think about this other idea, which is an effective population size, which is the number of individuals that are actually contributing basically genes to future generations. And we think, in wild populations, like, oh yeah, of course, they're all doing pretty well, they're healthy, they're surviving, they must all be breeding and reproducing. But what we know from other studies is generally that's truly not the case. And so when we go from one generation to the next, it ends up being that the future generations often only parented by something like 5 or 10% of the last generation. And so that's then gives us kind of an estimate of how healthy or inbred or uninbred or outbred populations are. So having those correlates gives us a way to measure or think about a population's genetic health. And folks kind of generally have some kind of hand-wavy ways of thinking about how census size and effective population size correlate, but we don't have a real good handle on how consistently some kind of back-of-the-envelope rules or generalities people use really hold up. And so that's something we kind of hope we can do, is because this has been a really long-term study, is look at both current effective population size and maybe potentially even what effective population size was like, 10 or 15 years ago, because we have those stored banked blood samples.
All right, that's really interesting. You can use genetics to actually get these ideas for the size of a population.
Yep.
What kind of markers do you use for that kind of thing? Or
You can use a lot of different types of markers. Generally, you want to use things that represent the nuclear genome. I've been doing lots of different sorts of genomic sequencing of turtles, both whole genome resequencing. So we're actually getting all 2.8 billion or so base pairs that exist in a turtle genome. And then there's other ways that we can look at reduced genomic sequencing. So we can use kind of some special lab magic to sequence the same couple percent of a genome across lots and lots of turtles, which also can give us a very good size of genetic health and things like inbreeding potential or depression and genetic diversity or heterozygosity.
That's really interesting. Yeah, you had one of the papers I think recently with the desert tortoise, the translocations. That's something.
Yep.
I mean, that's different. You were looking at heterozygosity, but
Yep.
kind of a cool tool. And
Yeah, but heterozygosity is generally an indicator of long-term genetic health, and so when populations, it's basically the inverse of being inbred. So when we're inbred, we lose heterozygosity through inbreeding, and so it's kind of that kind of tool. And desert tortoise thing was fun. We kind of found that those critters that had the highest heterozygosity have the highest survivorship if we think about moving animals around or translocating individuals from one population to another to try to augment population sizes.
almost, I mean, it makes sense, right?
Yep.
You've got individuals with the most variability genetically. It's just there's a chance that they're going to be more successful through the totality of sort of those effects.
Yep.
Yeah, that's really cool. I guess maybe the last thing we can touch on, we're just having a big discussion about this, but we'll get wrapped up, is the situation with the Pseudemys.
Oh, yeah.
And that was something you worked on, but maybe you can give us the rundown there.
Yeah, we need to get that wrapped up, but we did sequence about 80 Pseudemys genomes from all representatives, and between 5 and 10 of each kind of proposed nominal taxa, including subspecies, and we're kind of asking, how well do things like Peninsularis hold up versus Floridana or other subspecies of Cancina. There's been lots of described diversity within the wide-ranging river cooters, Pseudemys cancina, and whether or not any of those kind of hold up genetically. And then also looking at things like red belly turtles and the Texas cooters, Gorzugi and Texana. And kind of to summarize what it looks like the data are showing us is that Peninsularis is just a type of Floridana, or nested within that group of turtles, and also similarly Swaniensis is kind of nested within the larger Cancina clade of turtles. And so it looks like we kind of have a Florida cooter, a river cooter, 3 red bellies, and a couple Texas critters.
Could you make the argument that Swaniensis is its own clade within a clade? Is that something that...
In terms of species level diversity, folks generally aren't super fond of that. I think if folks like Swaniensis as a subspecies is described, if you have morphology that holds up, that or color patterns or chromatic variation that holds up, and you want to keep that as a subspecies, that would be perfectly kind of valid. But because it's nested pretty high up in the rest of Cancina, generally when we have relationships like this, kind of the two alternative approaches that are considered kind of kosher are either to subsume one group into the rest, or to make basically all the other populations or clades at that level their own species as well. And I don't think that there'd be lots of support for that within the Cancina. There's kind of lots of clades or subpopulations within Cancina that are all pretty much just geographically cohesive.
Right.
So like animals that live in similar watersheds are more closely related to each other than those from further away, and so it kind of makes sense from that biogeographic standpoint.
Cool. All right. Well, I guess we'll wrap this as a quick one. We'll add on, but thanks for sitting down with us.
Of course. Thank you. I appreciate it.
Hope you're enjoying the Florida weather. And
Florida's great. Turtles are all over.
Yeah. All right.
Cheers.
Thank you.
All right, this is the last part of the season 1 finale of the CheloniaCast. We're actually recording this more than a month after the first part. And as you can see, we've gotten a little silly just because this is our last recording of season 1. But we're joined by Gabe Weikert, who is the Herp Club. He's also pretty heavily involved with herp stuff, and he's all over the place. If you look on his Instagram, he's all around. So we're excited to talk to him, but about his experience at mostly at Rainbow Run, but just with herps and turtles in general. So thanks for coming on for a little short one, Gabe.
Yeah, thank you so much, guys.
All right. Well, I mean, so you guys were, so this was something we wanted to do kind of early on, like right after the first survey that we did, like late October. But just all of our schedules got kind of crazy. We're all in college now, and just kind of getting through that was a lot. But since then, you guys were literally at a survey there yesterday. Maybe just give us what were you guys finding, and what's that experience like in the water, kind of as a student?
Yeah, so yeah, I'll kind of give the spiel on what Rainbow River is, kind of why we do it. Dr. Meylan, you obviously talked to Dr. Meylan about it. Long-term study at Rainbow River, one of the larger spring systems in Florida. We have our typical, Consina, Swaniensis, and then we also have Nelsoni for our Pseudemys, and then we usually get Florida softshells. We usually get one snapper. We didn't get one yesterday, and then we get our musk turtles as well, Oderatus and minor. And I mean, basically, as Herp Club president, I just kind of facilitate the trip for everybody, make sure that we can get as many students involved as possible, as many new faces who have never done research work, and just make sure the trip goes smoothly and make sure that people know about it so they can get involved. So I'm just in charge of, like, if needs anything, I have to provide it, basically. So Dr. Meylan doesn't have to worry about anything except for showing up. Yeah. And I'm kind of a point man too.
Right. You guys, and what's the process for that? Like everyone shows up at Eckerd, because the spring is, I mean, it's kind of a haul from Eckerd itself.
Yeah. We kind of put together a pretty good plan on, like, we meet up at like 7 a.m. Everyone kind of carpools together. I had to lobby for a budget, money and pit tags and other stuff. So it's really nice that Eckerd does that we can have a club and get money from the school to pay for things. So we kind of meet up. Everyone drives to Rainbow River after I've collected all our supplies, and then we get to Rainbow, and like you guys saw when we were there, and anyone who ever comes another time, is we get there at 10, like around 10, and by 10 o'clock we're trying to get into the water and start our surveying because we have a lot of things to do that day. Thankfully, it can be a big survey, but we have so many people on turtles that, like, it usually goes pretty smoothly, and everyone gets to do a lot of important work. I think it's a great entry level, but also it's higher level because it's Dr. Meylan's long-term study. So it's cool that you bring in. We usually bring in people like you guys who know a lot, or Dr. Meylan. We just brought on Dr. Scott, and having all those people, but also there's kids who, when you guys were there and even yesterday, showed up for the first time ever. So I really like that about our surveys. Yeah, it's kind of how it goes down. We get to Rainbow, we do our surveying. By 2 o'clock, we're trying to have turtles going to X-rays. So it's just kind of like a flowing survey in that way.
Well, I remember too, when you were there too, it was impressive how involved you were in terms of like a leadership role. Like you were kind of running around getting people to do stuff, and that's a tough sort of thing to do. Is that something that's kind of always on your mind? Like, I need to be in these places to try to facilitate that sort of thing.
Yeah. If I mean, obviously people listening weren't there, but some of you guys saw, there's a lot of people are doing certain things, and I'm just kind of there to make sure that, like, oh, we don't have pit tags for the musk turtles. I go and grab some. I'm just like constantly thinking about what we're not doing, what we haven't done. That's kind of like the role I've taken in Dr. Gessling's lab. I feel like a lot of the time it's a lot of problem solving for just little issues that we run into. But I run into the problem where, like, we have people standing around at the survey, and we need to get out of there because we could end up being there for a long time. and people see me just running around thinking I'm just having a good time, which Jack knows, like, there's a reason I go over to that Sternotheris table, because we get done with it quickly and then we get to play with other turtles. But
No,
mind you,
get through that quick too. It's pretty impressive. Yeah.
But mind you, there's a lot of other things going on too. I'm trying to make sure we're at the vet's office in time too, because they're going to get annoyed if we show up at 3:00 and they closed at noon. There's just always silly things. And I've always been in athletics and been like a captain of my team and been like a leader. So like, it's been easy for me to take on that role. But yeah.
The sports connection is good. I like to tell people that too. Like, I played football freshman year. Jack did it, I think, the whole time, and it's just like that kind of thing carries over a lot to research, more than people think. If you don't get seriously injured, it builds a lot of good, I think, skills and like realistic leadership skills and just ability to kind of work as a team, that sort of thing. So that's an interesting connection.
I can definitely, I would say, well, there's a lot of like aspects of getting scratched up and bit by turtles, and teamwork and other aspects. And then also in the same way, like, I don't know, you got to be athletic to catch turtles. So I think that's something that's kind of missed on a lot of time. The kind of surveying we're talking about doing, you got to be able to swim, and you got to be able to swim faster and swim with soft shells and stuff. So I think it's a good connection I have. I actually started off at Eckerd playing soccer my first year, and then I quit to do turtle stuff. So
yeah. Well, speaking of soft shells and swimming, I think so you guys were there yesterday.
Yeah.
You got to have some every time you get in the water. For anyone that hasn't snorkeled for turtles, there's always, when you get in the water, it's always a different adventure. I mean, because you guys are in there for 2-3 hours. I mean, you always are finding something new. What was the story from yesterday? What was the coolest snorkeling find? Or
I think Jack's got this one, because I was freaking out when Jack brought his GoPro to me after.
Oh yeah, well, what he's referring to is, as we were leaving the survey, this actually isn't turtle related. It was totally. I had never seen a water snake actually in a spring run. There was a huge brown water snake just on the bottoms, coming up like not parallel, directly in front of me. And I just swam over and got my GoPro out, and I totally expected it to be fearful and try to get away. We do, but it comes right up to the camera and just like pokes its face up and follows me around. And when it went away, I just let it go. I didn't even try to catch it. I had too much respect for it at that point, because, but yeah, and no one was believing me. I started showing people the video, and they're like, "Holy crap, that's insane."
Yeah, I will say we also, it was a good survey yesterday, but we had a slow one. I will say, Dr. Meylan just sent me the numbers, and it was only 70 turtles, which if you put that into perspective, which I don't know if that's worrisome. On this date a year ago, literally today, there was 240 turtles caught. So I don't know if it was temperature. I think we debated about canceling last year's because it was so cold, which definitely the cold weather, I feel like, slows down turtles. They're more dependent on basking, makes us more likely to catch them. But big discrepancy between 240 and 70 in 1 year. It's kind of, and then, every survey since that 240 turtle survey has been slowly decreasing. So it's kind of something that, I don't know if it's because we're surveying again. We came out of COVID. That's why we did that survey last year. It was our first one out of COVID, but I'm still trying to figure out what's going on there. I really want to do a survey on like a 50-degree day and just see if we just catch a ton of turtles, and maybe that'll solve my problem that is continuing to perplex me. But it was still a good survey yesterday.
Are those numbers biased towards Emeritus? Like when you had like 200 plus turtles, are they mostly cooters, or has the Sternotheris level changed much between those two?
I can pull it up pretty quickly because I just typed in my notes app after every single one. But I will say musk turtles, it is like you have like when it's those types of days, you have like a few, I would say, buckets full of musk turtles. It's pretty crazy. So it looks like that day we had 122 minor and 23 odorated.
Oh, okay.
Yeah. And last year we did have a survey, though, that we got more consina than minor, 55 to 54. So usually it's just a ton of minor, which if people don't know, minor were introduced to rainbow like 70 years ago somewhere. So they're not actually native, but so they're kind of taking over the Odoratus.
That's interesting. You said the 240 turtle survey, that was right after, that was the first one after COVID.
Yeah, so there have been about, I think, a year and a half of no surveying.
Right, right. And yeah, so between now and then you've had, is that what, 6 surveys then, or 4 surveys?
1, 2, 3. We've had 4. Yeah. The thing that I will say is it was like 140, 130, one like 25, somewhere in there. And then this one's a big drop off, but it's 80 degrees in December. And it's kind of, I feel like that's kind of an outlier, and I feel like last year could be an outlier. So like in that way, it's like I still need some more weather differences, but it could be release strategies. I've thought about that too. There's a lot of different things that could be going into that, but I'd love to hear what you guys have to think about it too.
Yeah, I mean, I think it's interesting. It's like the only thing I think about is that you don't survey super frequently, which is not, I mean, obviously it's a tough thing. You understand it more than most in terms of the logistics behind it to coordinate. Even 2 times a year is that's a lot of work. But I would just kind of wonder, like, short-term trends. I mean, it's certainly something. It may be a mean swing, but when you're talking about like turtle population dynamics, it can take a long time for those trends to manifest. Like, I think about, and I mean, I think that as kids too, we have kind of a different, not maybe not kids, but younger
Mhm.
younger men, I guess. We have kind of a different perspective. It's like shorter term perspective, whereas a lot of times I feel like things maybe fluctuate. They oscillate a bit more than we think when you look at it long term. Like for me, when COVID hit and everything, I had all this big psychological bias. Like, oh man, the creek near me is just clear as day. I mean, this is just ridiculous. And then in hindsight, after going back for a few years after the fact, it was a really different, at least in the visible aspect that I was looking at than years after the fact. So I mean, that's just for me, that kind of stands out. And I was thinking about that even today, like, short-term trends are tough to make long-term forecasts, I guess, but it's something, right?
Yeah. I certainly, I talked to Dr. Gessling about it and Dr. Meylan, who have been on this project for a while, and they're just like, yeah, it's whatever. We'll see until we see like actual. And Bill Hawthorne, who used to be president of Herb Club, some of you guys have met as well. He also works for the Springs Institute and has surveyed there, and he said that it was something with the fish abundance or diversity was really low, but it is the lowest, it's the lowest it's been in a very long time. So I'm starting to put those things together, but at the same time, like you said, like it could just be, the weather that day, or who's catching turtles, or, what the turtles are doing. It's totally, we'd have to wait a lot longer to see if anything happened. But the difficult thing is sometimes we could wait too long and never know. And then there could be, so I guess it's, this or that.
Yeah, it was fun to be out there too. And I mean, yeah, in terms of the trends, it's just tough to predict. Like with the I mean, some of the published reports in like Cooters at Blue Spring on the Atlantic coast of Florida, there's a time with some of their models, there's a time effect. Even they survey, actually not too frequent either, but there's some level of like recapture probability has a time effect to it. So it seems like maybe certain individuals are in there at different times, and it's just kind of tough. But yeah, it's cool to just, I mean, any trend is a trend, right? And it's cool to see how it adds up over time.
Yeah, so I just pulled up this. You know how I alluded a little bit to the musk turtles kind of going like this. They kind of did this over the last, it says from since like 1985, but there's been a like literally it's been a trend one way and the other. Miners going up and Odoratus is just going straight down, and that took at least like 50 years to figure out of data. But it's pretty clear now. There's like an 8-to-1 ratio of minor to Odoratus when Odoratus used to be 100% of the population. So but
That's
that took a long time too.
that's
Why do
still
you think miners are more successful in this habitat than Odoratus?
Um, you know, I haven't really thought about it that much. I mean, what do you think? I think that, minor are just naturally a more spring going species that you're going to find. And I mean, you think about all of our Santa Fe stuff that if you guys have ever, been to Santa Fe, it's like Ichtuckney is full of minor. Obviously I'm pretty sure they're native there as well. Yeah, I've talked.
Yep.
Yeah. So, and then Odoratus are just, I feel like they're just smaller there at Rainbow. So you can definitely see that. It's very, they're plastic. So in Florida, they're a lot smaller than up north where they get a lot bigger. And so I think it's got to be size, or maybe the niche that they're filling. Could be a lot of things, but I would say,
it's probably not done either, because if you look at like Ichtuckney or other springs where loggerheads are native, and so are Odoratus, you tend to have a really low, like a crazy ratio of like Odoratus to minor. It's like one Odoratus for every 50, 100, 150 minor. Like I think it's probably going to keep going down if it follows the trend that you see in other springs. But overall, it's probably just other forms of competition that I would think. But
Is the abundance of minor in there, is it at the same level the Odoratus were at prior to the minor being introduced, or is it surpassing even that?
Well, what's interesting is, so I've been told by Meylan and guessing that they were physically introduced by someone who used to be a biologist, but they were actually a poacher who kind of just did what they wanted. And so there was no minor at all. And so to that point, I mean, I want to see what year the data started, but at some point they got introduced and there was none whatsoever. And then it's just been slowly climbing up. So maybe there were some who just didn't know because they weren't surveying it. And maybe it's just trends with the water and stuff. And maybe that's what's happening, like we see in other springs. But I would say we won't know, because you would never know if minor were. I mean, I don't think people were surveying the river back then. So, really,
Right. Yeah, that's a good point. But like looking at the trends over time, it's certainly kind of climbed. That proportion has changed, and too, like if you just had a 5 or 10-year snippet, you might not think anything's going on. Like with turtles, turtles take a long time, so but it's cool that data is there to begin with, and it's even to some aspect, it's even cooler the fact that so many students have been involved with collecting that over that amount of time. And that not only is the data there and it's really impactful data, but it's also had an impact on, at this point, probably hundreds of biologists that are kind of the future generation. That's a cool thing.
Yeah, I kind of sell it as that to a lot of people. I really try to get to like freshmen and sophomores. I go into our introductory bio courses and tell them about it, because I mean, this is the best way to get into it. It's the best way to start working with Dr. Gessling. I didn't start this way, but I know a lot of people did. A lot of theses have been done on this project with specifically the Odoratus and the minor. But, I mean, we have a student, Jorge. Well, he's no student, but he went on to master's, and now he's going to, I think, Utah State for his PhD, and he did his thesis here on the musturals as well. So the projects have provided as well for our students, which I think is awesome. And it's going to be great for me too. I got to meet you guys and work at Santa Fe for the last few weeks with Jack, and I saw Michael there too. And I plan on keep on doing that and just getting involved more with turtles, because like I told you guys before, like I was doing gopher tortoise research all the time. I love gopher tortoises, but they're not like my calling either. I wouldn't say like I love them to death, but I like to swim and catch turtles. So.
Yeah, that's a different thing.
something about the swimming is just really totally different than like hiking or something. I don't know. I like it a lot better, if I had to be honest.
Right. Well, I think we're starting to come up here, but I just for Gabe, I'm curious, like what are you thinking, like future plans? You have career wise or?
You know, really to like be very like I don't know, I guess presumptive about it. I would really like. I mean, I would think that maybe a PhD is somewhere in there, pursuing grad school and all that stuff, and maybe becoming a professor and doing research. But I don't know if that's me completely. I'm not really. I like to. I'm more like someone who enjoys surveys a lot more than sitting in the lab and writing a paper. So I don't know where it's going, but I also got to maybe I'll find that in the next few years. I've jumped right into things from my freshman year doing research, and I feel like now I can finally slow down right now and trying to figure out what I want to do. But otherwise, outside of that, just do turtle research wherever it happens, whenever it happens. But I'm just going to continue to make connections with people like you guys, and hopefully, I'll find my place somewhere. But it'll probably be with river turtles. I would really hope so. Maybe up in Ohio, where I'm from, with some terrestrial turtle species. But I'm not really sure. It's just I'm going to be working probably with Dr. Gessling over these next few years. So maybe the gopher tortoise stuff will take over, but I kind of hope not, but we'll see.
That's cool. Yeah. I feel like for all of us too, it's like a lot of people, I don't know, they probably realize, but a lot of people just look at us like we're all interested in that sort of thing. And they think, oh, these guys have it figured out to some extent. But realistically, like career wise, I don't know if any of us know like exactly
No.
what we want to do.
I'm in pretty much. I'm in a very similar boat. Like the only thing I want to be involved in. I want to be heavily involved in turtle research and pursue. I want to go to grad school, but like I don't have much anything else dotted out other than that.
Yeah. Plans to buy a house somewhere on a river, somewhere in the south.
Oh yeah, that actually is something.
Every morning.
I was planning on buying
or whatever.
Yeah.
Planning on buying
with permits.
Yeah, well, yeah. I'd love to just buy a piece of land, even with no house on it, and just live in an RV for a while and eventually build something. But just to own a piece of land or something. I don't know. That's pretty off topic, but
no, it's not. But have you? Yeah, sorry, never mind. I don't know. I was going off in a distant place. I was like, what, do we still want to keep talking about turtles, or?
No, I mean, I think
No.
we can wrap up, but
Okay.
yeah, I mean, I have one more question actually. I'm curious. Because when I first heard about Eckerd a long time ago from someone, and I went to North Carolina and did this like study away thing, and it was like to me that the turtles were like he told me everyone knew about the turtles there, and that was kind of a big thing. I'm just curious what is are they like, is that project kind of a famous thing at the school? Does everyone want to participate in it, or how does that work?
Well, I've talked to Bill a lot about this. We definitely have a good group of herpetologists here, a very good group, I would say. Dr. Scott has done awesome things with intermediates, and then also desert tortoises, and Dr. Meylan's always been doing his sea turtle research. I know his sea turtle research that he's been doing with his wife is massive. I don't know really how it's related, because I don't know much about sea turtles, and I don't talk to him about that stuff. I talk to him about Rainbow River stuff, but I know the Rainbow River turtle survey is one of the longer ones that's still going on today, where students are involved in the aspect the way they are. This campus is a bit like, it's just a lot of animal people, big marine science school, big biology school, environmental studies. If you go around anywhere at night, there's, I would say any given night, there's kids catching snakes in ponds or whatever. Like this school, it's very, I don't know, animal oriented. Like everyone's allowed to have pets and stuff. We have a chameleon. I've heard stories of people having diamondback terrapins and stuff. So like it's just like an animal school, and there's a lot of people who care about reptiles here. And so that's how like Gessling got here as well. and we kind of cover all aspects of turtles: sea turtles, tortoises, and river turtles. So I don't know. It's just a turtle school. That's all I have to say about it, I guess.
Yeah, it is a, from all accounts, and that's just another one. It's like it's a, to study at, and just to, I mean, the campus itself is incredible. So that's for anyone that's younger and interested in pursuing biology, definitely check out Eckerd. I think it's a great place. So, but all right. I mean, I guess if anyone else has anything, but we can sign off for now. But
Yeah, thank you guys for having me, and I'm sure I'll see you guys around pretty soon or in the next few months.
oh yeah, we'll be down there next month, right?
When do you
Yeah.
head off, Gabe? You said.
January 3rd, but I'll be gone for winter break too, or for 2
Oh
weeks.
yeah, that's right. Yeah.
But I know we talked about, doing stuff next spring, so
Yeah.
hopefully turtle stuff to come.
For sure.
All right, sweet. Well, this concludes the final episode of season 1. We will have season 2 ready at some point. But thanks for listening to this first season of CheloniaCast. Leave a review, drop a like, subscribe, do whatever you do on Spotify, Apple Podcasts. But all right, thanks for joining us.